So, hi everyone, we're ready to start. And we're happy to welcome you here at the public health hackathon organized by IBRI in partnership with Kazakhstan Sociology Lab. And before we officially begin, before the official opening of our hackathon, we have a pre-start lecture for you dedicated to machine learning in genomics. And I have the honor to introduce the lecturer. So please welcome Nikita Vaulin, bioinformatician, SchoolTech graduate, and PhD student in Medical University of Vienna.
Yeah, hi everyone. Really great pleasure to meet you all here in our team. And let's start with a small warm-up before the hackathon begins and discuss some machine learning. You know, machine learning draws a lot of criticism in different fields, but at least there are some places who believe that criticism is not that much like pronounced and there is still some hope inside. So let's talk something about that. I was already introduced by Nadia. Thank you, Nadia. But at least if you want to contact me, you also have my contacts and feel free to text me anytime you want. And the point of why I selected to this topic is because 1 month ago, something happened and we will discuss what happened 1 month ago. Then we will discuss which people do this amazing things. And then we will discuss what we can get of it. And what happened, it was, where should I put it? Okay. Oh, great. Yeah. And also disclaimer that there are a lot of machine learning like people discuss this is like bad machine learning or whatever, like foundation models as CGPT. We will not discuss it today and we will not discuss it here. So there will be another stuff. And another stuff is the model Alpha Genome that Google released approximately 1 month ago or 2 months ago already. And this is from one point of view. Someone said like this is another breakthrough like AlphaFold, and now we have Alpha Genome. Other people say that it's not a big achievement nowadays. So let's take a look at this thing closer. Primarily, what does this model do? It takes just DNA sequence alone, and then somehow it transforms it to the numbers, which are just functional profiles of whatever you want. It can be RNA-seq coverage, or ChIP-seq coverage, or ATAC-seq coverage, or whatever. And then this is basically a huge tool that you can use to make different predictions across different modalities. And there are really a lot of modalities. Most all of them are listed here, and they can do it even with high C maps or other functional profiles. And the resolution also is really great because they can do RNA-seq coverage just with one base pair resolution. So for each nucleotide, you know how much it is expressed, and you don't need to do any experiments. You don't need to go to the wet lab. You just open your notebook and you upload the DNA sequence, and then you get the predictions. Yep. But then also what they released preprint for 100 pages, and they also provided you with IP so you can play with this model. And also you have a forum to chat with other researchers who do some research and who try to run this model. But main thing is they didn't provide us with the model itself yet because it's still by archive preprint. So they plan to release the model weights when they will have the publication. So you can reproduce the results in the preprint, but you cannot do it on your own data just for now. So you need to wait. The same was with AlphaFold and with other models. But this is the comment of one of the developer of this model that they will release the weights upon the final publication. But then also who did this research, and two persons that are really important in the machine learning fields are the heads of the DeepMind Google, Pushmet Kohli and Demis Hassabis. And also Demis Hassabis, this is a guy who was awarded the Nobel Prize the last year for the AlphaFold development. And all of them, like they leaded all other DeepMind products. But also, like the actual work was done by the group of the ML engineers and like kind of postdocs or PhD students, but they're in the industry. And one of the guys, a lady, Natasha Latisheva, she is Russian, but also Zhiga Ovsek and Kyle Taylor. But all this work was not like, they was not the only guys who were working on the ML field. And there is also another group that I like want to introduce to you. This is a Kalika Labs, also somehow associated to Google, but this is the guys who were doing this machine learning research really like from 2016, and they were like training all the models, and then DeepMind appeared in this field much later. And David Kelly from Kalika Labs with his postdoc Vikram Agarwal, they did a lot of research connected to both machine learning and to some dogs, because they were naming the models based on the dogs, and all of the tools somehow related to the breeds. And then all this like, I want to show you that all these huge achievements in the science, they appear actually like being incremental with small steps. For example, for Kalika Labs, they firstly started with Basset, and they started with DNase activity, just one track, just one prediction, not RNA-seq, not ATAC-seq, just DNase1 activity, and they did just binary predictions. So they didn't provide coverage. They just answered, is it bound by DNase1 or not? And that's it. There's nothing more you can get. But they already like played with deep neural networks and showed that it can be useful. And then they did like these incremental changes. This is the part from the paper about how they named the thing. You can read it later. And then they like transferred to another model, Basenji, which was like able to provide already qualitative predictions. So not is it like bound or not, but already some kind of numbers that somehow correlate with something. And those numbers were also produced for CageSeq, which is basically expression, and some Houston marks. And this was already cool. But the next huge step was 2 years after with Basenji 2, where they did pretty much the same, but they provided a new concept of cross-species learning, because all of the work previously was done on humans, but then they showed that if you train a model on both human and mouse data, which are completely different organisms, but if you do it on different organisms, that somehow model appeared to be better. And this is also a cool result, that you can integrate really, really different things, and somehow you can get better results from it. But then you get a really, really important notion of you need to be careful while doing your train test split. And for validation, if no machine learning, then you can think about what can be wrong. And the answer is that you have different organisms, mouse and human, and they have orthologous regions. So you don't want to be the same region from human in training set and for mouse in testing set. And you need to be careful about that. I will not introduce like to the reasoning itself, but there is like a lecture in Russian, I'm sorry, by Vovashitov, who was discussing also some problems about train test split and the machine learning. And what authors did, they did really careful not separating orthologous regions into different data sets. So there is some like graph-based data science, but we don't know to discuss this. We don't need to discuss it right now. But this is an important thing to keep in mind, that you always have some limitations and you need to be careful. And if you don't know about the limitations, then you need to spend some time on exploring them. And then also they played with other models, they played with other modalities, they started to predict at high C, and this is really like much more hard data because it's two-dimensional, not one-dimensional compared to other modalities. So this was another huge step. And then all of these things collapsed into the one model, which was done based on the really cool concept that already changed all the machine learning field, which is called transformer architecture. And the paper called Attention is All You Need. Again, Google provided us in 2017 the architecture transformer that's already used in many, many different models. And this architecture was used in another model informer where Kalika Labs and Google DeepMind Research, they like kind of worked together to do some like another huge achievement in the field. and another guest for the AlphaFold. And here they did like more modalities with high. And this is really important. The window size matters because you want to kind of track the interactions between different genomic regions. You want to see that some cancer activates some gene or whatever. But then if your model can take into account only like 10 base pairs, then you don't see much. And if your model can take into account like 200 kilobases, then you can see some distal regulatory regions, distal regulatory interactions. And this is what all this field is trying to understand, trying to explore, and to describe. And then another like huge but incremental step was developing another model, Barzoi, which was provided by Calica Labs alone, where they did much more important focus, where they made much more detailed focus on the RNA-seq analysis and RNA-seq predictions. And then like all these models, as I said it to you, they take DNA sequence alone, just letters, and transform it to numbers. But there was a big problem with predicting RNA-seq. This is why I mentioned it in the slide before, because you can predict chip-seq, you can predict cage-seq or attack-seq or whatever, because those peaks are relatively small, and it's easy to like provide 10 numbers or 20 numbers. But if you want to do RNA-seq, this is like the genes in human genome, they're really long, and you need to provide predictions for huge amount of input data. And then models appear to be less effective with a very like broad range context, and you need more computational resources for that. So it was a big problem, and people tried to solve this. And the final aim was to predict RNA-seq, and it was kind of solved already several years ago. But then this is a model Barzoi, yeah, and this is like kind of predictions made for some particular tissue. This is the actual RNA-seq coverage of some particular gene, and you have introns and exons marked here, and you see the Barzoi predictions below, and you can like understand that maybe it's kind of good, and it kind of can like reproduce the actual data. Maybe it's good, but why don't we need it? We'll discuss it later. And why it's good, this is another thing I wanted to mention, not technical detail, but ideological detail. There was a problem that you need to work with a lot of data, and they were like trying to find different solutions. And then they find that in image analysis, there was a funny trick, which is called U-net architecture, where you do this like some machine learning magic, but then you kind of multiply different layers from the beginning and the end of the model, and then it can provide you better results. So this is the thing that was already known and vastly explored in the image analysis and all the AlexNet like things that were after AlexNet and image recognition. They were like mainly also done because of this architecture, and then they applied the same concept in the genomics field. Yep. But then also there was some kind of weird thing, because for example here just screenshot from the paper that how they did normalization. The y is just counts, and if you want normalized counts, then you do this kind of to the power of 3 or 4. But if it's less of some number, you do another thing. So sometimes people in machine learning field they do strange moves, but this is also how the thing works. Yep, and then we have AlphaGenome. And it can provide, as I said, like more modalities. It can provide more resolution, up to 1 base pair, and with much broader window. And this model, it's already like integrated all these incremental achievements in it. It used multi-species approach. It used transformer architecture. It used U-net architecture. So this was like a collection of different achievements people discovered on the way. Yep, and this is kind of examples of what you can get from it. For example, this is expression predicted by AlphaGenome, and this is some gene which is important to be expressed in skeletal muscle, and you can see that there is some expression on the skeletal muscle, but it's not expressed in other organs where it should not be. And you can compare the observed and predicted expressions. And also you cannot like predict on the level of the whole gene, but you can predict specific isoforms. For example, here you have difference in the 3' UTR region, which is like here's the prediction from Barzoi because I had just a nice picture. But you can do the same with alpha genome and all other models of the same age. And also, like here appears, can you tap please? Yep. But here appears a big problem, a big question. Is like, as I said, these models, they can kind of predict your coverage. But in order to train them, you need to have this coverage. You need to do an experiment. You need to provide the actual data. Then you show it to the model. Then model trains, and then models is like really successful in predicting the things you already have. And the question is like, then why do we need those models? Yeah, why do we need them? And there comes some kind of not machine learning, but scientific research of what can you get from the stupid ML. For example, what you can do, you can run in silico experiments. So you have your DNA sequence, you can predict the values. You did it for some like reference species, and then you can try to mutate the sequence, and you can try to see what changes it makes. You cannot transfer these predictions like to the clinic immediately because this is just some numbers from some computer. But then you can use it as like to narrow the window of the candidates that you will put into the actual wet lab experiments. And there is like whole procedure which is called in silico mutagenesis, and you can like, as authors say, you can call different quantitative trait loci, different QTLs for different traits. And you see, for example, here they introduced some mutation in some specific gene, and then also you have the blue is reference and the red is mutated track, and you see that the supply site was introduced. And in the paper, they show that it's some kind of related to some specific some specific parts of the biology that were not discovered yet. For example, there was some gene that was like appeared to be oncogenic, and no one knows why. And then they did some experiments, and they showed that if you do this mutation, then you introduce MYB by the site, and then something bad happens. So this is kind of research you can do with it. And also you can try to understand model reasoning. So if the model is really good in predicting the actual coverage, then you can try to understand why is it good? Why, like at what things it's looking at? And this is kind of like a simple scheme. So if you simplify the cell, so cell is just a machinery that transforms DNA sequence into the functional profiles. And our model is also some kind of machine that transforms DNA sequence into functional profiles. And if they do it the same way, if you understand how model does it, you can understand how cell does it. And this is kind of the concept that you can solve with different approaches. And the most like well-known is called integrated gradients. Doesn't matter, but you can try to understand importances of each particular part of the input data. It's really like famous for the image analysis. For example, if you want to classify this image of which animal is on the picture, you can do it, and then you can try to understand at which pixels model was looking in order to provide predictions. And you can see that actually it's also meaningful, and the model is looking like at the animal itself. And then you can do the same for our genomics models. But here in the input data, we don't have like pixels, but we have DNA sequence. And if some nucleotides are really important, so on the y-axis here is like importance, then if those nucleotides are really important, maybe something like happening there. And yes, it appeared to be like motifs of some transcription factors. And you can see this effect's really far away. So there is some gene MYC, its expression, and then authors find some way in this region, many kilobases apart, some like motifs of some transcription factor appear to be important to provide a prediction that this gene is expressed or not. Another concept is directly connected to the transformer architecture. We will not talk about architecture in detail, but just in simple words, it's some kind of weird matrix multiplication inside. And those like matrices, they come from the DNA sequence. What does what this machine model does it provides you like some hidden representation of some like inner matrices. And then from those matrices, it provides you a number like 42, the expression of particular gene, let it be 42. But then this multiplication inside, it goes that way that different parts of your input data, it like somehow got multiplied by each other. And then the funny trick comes out that it means that this is like answer is 42 if X is equal to this sequence, but only when Y is equal to this sequence. And if you change one, the prediction also will be changed. And then you can kind of assume that if both two regions are important to this prediction, maybe they some kind of like transcription factors can be bound there. And if the result is this multiplication is really strong, if it's really high, we can assume, we can propose that those two transcription factors, they actually like interact with each other. And this is what you like obtain as what is called attention matrix. And for those who do high-C analysis, it can be like look similar to high-C data analysis. But yeah, so here you can see some bright pixels. They mean that you have the same genomic region on the x-axis and on the y-axis. And if the pixel is bright, it means that those two regions are important for each other. For example, this thing means that those two, like some particular parts of input data, they kind of interact in the not in the spatials, like in the spatial modality as high-C, but in the like semantic modality, like in the model. And you can like see that some of those like stripes, they are kind of related to splice size or whatever. Yep. So this is like kind of the summary of what I talked to you during this, I guess, 20 minutes. That this is just like, this looks cool, but this actually small step in this field, and it has a lot of like drawbacks. First of all, it actually did a lot of mispredictions. I didn't show it yet, but they have in the manuscripts a lot of examples where the models like provides the wrong predictions. Also, like the input window size, 2 megabase pairs. This is a big size, but it's not the whole genome, and it's still some like space for further research. And also, it's really far away from personalized and single cell predictions. And even for other cell types or organisms, this is a very also key idea that you have this like in distribution and out of distribution predictions. And if you have pictures of animals and you want to classify the monkeys or cats, and then you try to recognize humans with these models, like to name the person, model will be wrong all the cases because it was never trained to predict humans. It was trained to predict animals. And here the same, if you like was training this model on some particular cell type on the brain cells, you can never rely on the predictions that were made for liver or other organisms. And then it's also like applies for the mutation tests. As I said, like you can try to introduce one mutation and you can see what it changes, and you can use it in your own research. But then if you introduce too many mutations, then it will be already like another sequence, and you never know where's the border. Where is like I can mutate one nucleotide, I can mutate two nucleotides. Where is the borderline where I can no longer trust the predictions of the model? And this is like the thing that people discuss in the like on the conferences or whatever. But then you can try to do what you can try to do. You can actually try to apply those models in your like I don't know life. First, you can just try the API. It's like relatively easy to start. You just need to generate the key, and then you can just follow the instructions. You can try to like play some mutations. You can check gene of your interest or another region or something like that, and you try like to switch it off or switch it on in the wrong cell type or whatever, and you can try to identify motifs that like related to the expression of your gene of interest. And also you can try to fine-tune this model on your own data, so to make it like more close to your research. This is not like something you cannot do with alpha genome yet, but you can try to do this with Informer and Borsoi. And this is also like some point that I would like to highlight right now, that these guys, all of them that I mentioned earlier, they really did a great job on describing how you can use their developing like their things, on how you install, how you run, and the documentation of all the things. This is really good thing that you can look at if you also like develop something. You can check how people from Google or Calico describe their work. Yep. And also, if you want to to get into more in more detail to this field, there is, as I mentioned, lecture by Voloshytov in Russian, but also there is Natasha's guide about the alpha genome, and also I want to introduce you to the IBRE Bioinformatics Club, which is also related to the artificial intelligence in the bioinformatics, which is happening right now, and you can also check what we're discussing there. And also there is some lecture in Russian from Blastim about the alpha genome, but I would rather read the whole paper or just pay attention if you're really not interested in this field. Yep. So key takeaways from all this topic is first, you can really find something interesting somewhere far away. You can look at the image analysis or some other fields to search for the tricks that can be useful for your own research. Another point is we are doing hackathon, you want to provide solutions or whatever, but then you always need to be careful with the methods you apply, and they also have limitations. And actually talking to people on the hackathon can be useful for you to understand, maybe someone knows that you cannot do something or you can do something. And this is also important. And I was introducing to a lot of people that were interacting. So the key takeaway is interacting with them. And so communicating and collaborating is really good thing. So that's it. And here I'll be happy.
Thank you very much. So we're ready for the discussion. If you have a question, please raise your hand, and I'll give you the mic. All right. So thank you for your lecture. Yes, something I didn't know.
Yeah, I don't know how to describe it in a good way, but this map, here you have some genomic regions, and here you have some genomic regions. And if the pixel is right, it means those two genomic regions, they interact. And this is true for high-C data analysis. They interact physically. But the problem is that here you don't do any physical experiment. This is something model imagined inside itself. So they interact, but they interact not physically, but I don't know how to say, a model thinks that they both talk to each other. It doesn't matter how they talk, but for models, they both know each other, they are friends. I hope I try to answer, but if not, we can try to discuss it later. But yeah, this is the hard stuff.
Does anyone else have questions?
Nadja?
Thank you for your presentation. I have a question about alpha genome. I didn't get a chance to read the paper in detail yet, but do you know if it's based on some annotation, official annotation of the genes, or is it sequence based? So because the annotation of the genome, we know that it's not perfect, and is it possible to say note down some arbitrary sequence that is not marked as a gene in the official notation, but possesses the features of a gene, for example?
Yeah, thank you. This is a good question. And the thing is, this model, they rely only on the DNA sequence alone without any prior knowledge about the sites of the binding or whatever. And for example, you see that for some gene, you have expression peaks on the exon or on the exons part. And then it means that model somehow it was given just letters, and it evolved the concept of gene, evolved the concept of the transcription factor binding site, evolved the concept of splice site inside itself. So it's kind of introduced the concept of splice site because it needed it to provide a good predictions. So we didn't know the annotation, but in the end, it provided you the annotation of the genes, not the names, but it found the genes. Yeah.
I know it's about a question from there.
Thank you for the question. I have a few questions in regards to numbers of accuracy of this new model. What are exactly the odds of mistakes or mishaps that can happen just that we cannot predict that? The process of working this model.
Yeah, it's a really good question, and I don't know whether I have something in this regard on the slide, but let me just open some picture. Yeah, no, maybe this one I want. Yeah, so they tried to evaluate the model because they had like the real tracks from the sequence experiment, the observed ones, and then they had the prediction tracks, and they like did this like most often train test split is done in that way that you have like 10 chromosomes left for the training and all other chromosomes are left for the validation. And then you kind of like try to correlate those two tracks. You can do just like Pearson correlation and you see how well they correlate with each other. And like on average, the Pearson correlation between prediction and the observation is like 0.95. So it's kind of looks good. And this is also like cool thing that this is including all the DNA. And if you focus on only on the genes, then they publish that the Pearson correlation for the genes is like 0.98, and then all the bad correlation is found for the region like the low correlation values are for the regions like far away from the genes, for the non-coding regions where I guess even cell doesn't know what's going on there, so the model doesn't know it as well. Yep. So this is the answer to your question.
I see. Thank you. But I thought that if the model rely only on the name and we have RNA coverage prediction, so it means that environment doesn't influence on this behavior. I mean, it's strange that we can see any environmental influence on our data, or even I also thought about it, maybe our difference between this model prediction and reality is maybe just environmental influence.
Yeah, the question is like the question about that we use only DNA and we don't account for the environment is really important. But like I guess the answer is that you don't train this model for the whole organism. You do as shown here for different like organs, but then you can focus on specific cell type and some particular place of the organism, and you do like kind of separate model for the neurons of the cerebellum, and you do the separate model for the hepatocytes, and you do the separate model for skeletal muscle cells, and then you like kind of assume that they have different environments, and it's kind of like you kind of have this information in the model that those cell types are different from those cell types, but you cannot like take it out easily, and you cannot like say to the model that we have this particular like morphogen floating around the cells. You cannot show it to the model, but you can show to the model cells with this morphogen floating around and without this morphogen, and then model will have to understand that something is like different between them.
Any questions? No, okay. I think that I have a question as well. Don't you think that even the implementation of machine learning methods in genomics, the future researchers' role will be reduced to just new model development and the old model results validation?
This is always like a good question. Will AI, like people say that those machine learning things that are just tools for us, but I also like have another question in my mind for me. Oh, another answer that you kind of with this, for example, if we imagine the best machine learning model ever that can like answer all your questions, I guess science is not about answering the questions. Science is about asking them. And if all the questions will be answered, then I guess scientists will still find another thing to research, because We are not someone who provides like the answers. We are the people who just wondering, and we enjoy